Okay, welcome to the video for chapter 5.4. 5.4 is all about where bacterial cells store their DNA. So we're going to talk about the nucleoid and we're going to talk about how bacteria divide themselves to make more bacteria. Okay, our section objectives for 5.4 are you need to be able to describe how DNA is organized within the bacterial cell, aka where can you find it, and you need to be able to explain how DNA replication is coordinated with cell growth and division. Okay, so if you remember correctly, prokaryotic cells have a defining feature. They do not have a nucleus or membrane-bound organelles. Okay, so where do we find bacterial DNA? Well, in bacteria, their DNA is found in a region of the cell called the nucleoid. Now, the nucleoid doesn't really have a specific place. It's just going to be wherever the DNA congregates. Okay, so just a reminder, bacterial genomes are one single circular double-stranded chromosome. Okay, so what's going to happen is this chromosome is going to loop together. So it's going to loop over itself and it's going to compact itself enough that it's only going to be found in one area of the cell. Now, the loops are it's able to stay looped around each other because of DNA binding proteins. It's going to help keep it into that nucleoid region. Now, the nucleoid region doesn't have a specific part of the bacterial cell. It's just going to be free floating wherever. It might be towards the center, it might be towards one end, but it's going to be where a majority of the bacterial cell's DNA is going to be found. Okay, but remember, the important thing is do not confuse the nucleoid in a bacteria cell with the nucleus in a eukaryotic cell. Okay, remember, bacteria have no nucleus, they have a nucleoid. Okay, so it's just where the DNA is found. And remember, it's going to be looped over each other. So we can kind of see these DNA binding proteins, they are going to help keep the chromosome looped so it doesn't take up as much space. Now we'll come back to what the DNA origin of replication means. Okay, DNA domain, this is just where it's going to be found in the nucleoid. So we have this single loop as part of our circular chromosome. Okay, so we'll come back to this. Now, the fact that bacteria have one single circular chromosome means that it's really easy for it to copy or replicate its genome. Okay, so bacteria are going to reproduce by a process called binary fission. Okay, so it's going to divide into two equal and genetically identical copies. Okay, we call these identical copies the daughter cells and the original cell the parent cell. Okay, sometimes binary fission is referred to as clonal reproduction because we're literally getting clones or identical copies of the original cell. Okay, so the process of binary fission is pretty simple. Okay, we'll also talk about this in more detail in a coming lecture. We're going to talk about DNA replication in itself. Okay, but in bacterial cells, we have our cell wall, we have our cytoplasm, and we have our chromosome. Okay, so this is our parent cell. So the parent cell is going to grow, it's going to start to overcome that surface area to volume ratio, if you remember that from biology. Okay, so when the volume gets too big, it doesn't have enough surface area to bring things across the membrane in terms of nutrients and to ship things out in terms of waste. Okay, so there comes a point where the bacterial cell gets too big to support itself. Okay, when this happens, the cell is going to start to elongate, or depending on the cell, we'll talk about this in a second. So the first step is when a cell needs to divide, when it's getting close to having a bad surface area to volume ratio, the first thing it's going to do is replicate its genome. So it's going to copy its DNA. Okay, so we have two copies. Okay, the DNA is going to get segregated, so it's going to move towards different parts of the cell. And then the septum, or a new cell wall, is going to, and cell membrane are going to start to be made in between the two halves of the cell. Okay, so this new cell wall and this new cell membrane are going to be built. This is what we call the septum. And then after that process of septation happens, we're going to get two daughter cells that are going to then go and grow until they hit their bad surface area to volume ratio, and the whole process is going to start over again. Now, with bacterial cell division, all the steps are quite the same between all bacterial species. We have DNA replication, we have a period of protein synthesis, so we have enough proteins to support each daughter cell. We're going to expand the cytoplasm, start to make more of it so that each cell can divide it equally between itself. Okay, and when we start to do this expansion of the cytoplasm and this extra protein synthesis, this is what's going to cause the cell to elongate. Okay, so once we have the DNA replicated, we have enough things to make two functional daughter cells. This is when septation, or that process of division, happens. Okay, so the septum forms and divides the cell into two. Okay, so we can see here in the micrograph, we can see the septum starting to form here in A. Okay, so it's going to start to continue until we get two different compartments. Okay, so this is going to be one daughter cell and this is going to be the other. Okay, so these are going to start to peel apart. And when they peel apart, the septum is going to completely disappear and we're going to have two daughter cells left. Okay, so this origin of replication, we'll talk more about it in the next chapter not the next chapter, in the chapter where we talk about DNA replication. This is where it starts to copy itself. So we're going to have one set of proteins go this way and this one set of proteins go this way, and we're going to end up with two identical copies. Okay, we call this the replosome. This is the thing that's going to copy it. Okay, this termination sequence right here in red is what's going to stop that replication sequence. Okay, and when that happens, we're going to have two identical cells after division. Okay, but let's watch a video on cell division real quick.
Cell division requires highly coordinated growth and expansion of all the cell's parts. Most bacteria have a circular chromosome that begins to replicate at its origin. At the origin, the DNA double helix begins to unzip, forming two replication forks. At each replication fork, DNA is synthesized by DNA polymerase molecules, which replicate the original DNA molecule, forming two copies. Replication proceeds in both directions around the circle. In a rod-shaped cell, the cell envelope and cell wall must elongate as well to maintain progeny of even girth and length. Growing bacteria replicate their DNA continuously with no resting phase. Sometimes even before replication terminates, two new replication forks form at each new origin of replication. Replication of the DNA termination site triggers growth of the dividing partition of the envelope, called the septum. The septum grows inward from the sides of the cell, at last constricting and sealing off the two daughter cells in a process called septation. Envelope extension and septation require rapid biosynthesis of all envelope components, including membranes and cell wall. Cocci, such as Staphylococcus aureus, do not elongate their cell walls before septation. Spherical cells expand their walls during septation. Here the furrows appear in the cell envelope all around the cell equator as a new cell wall grows inward. Two new envelope partitions are complete. The two daughter cells peel apart. The facing halves of each cell contain entirely new cell wall.
Okay, so in that video, we saw both the creation of rod-shaped cells and cocci-shaped cells. Okay, so when we talk about bacterial cells and what we see underneath the microscope, we have to talk about their orientation. Okay, so what are we seeing? Are we seeing chains? Are we seeing clusters? Okay, are we seeing individual ones? So these different arrangements of bacterial cells are all due to when and how they go through septation. Okay, so when it's going to make a rod-shaped cell, okay, we saw that elongation happens at that protein synthesis and cytoplasm production stage. So it's going to start to elongate to fit everything in the cell. Okay, so rod-shaped bacteria, they elongate before they go through septation. Okay, cocci, on the other hand, don't elongate. Okay, so they stay spherical. Okay, so they keep individual circles and not the rod shape. Okay, so we can also have an arrangement so both the rod shape and the cocci can make chains, so more than one cell connected to each other. Okay, so kind of like if we look right here in this, we see that it's a chain of cocci. Okay, but what about these little bunches? We call these staphylum. Hopefully I can say it. Staphyl. Okay, staphyl looks like clumps of grapes. Okay, in order to get clusters instead of chains, what's going to happen is when septation happens, instead of happening at the same orientation, so at the same place each time to make the chain, it's going to alternate. So this is my cell, and say that I copy on this side of the cell. So septation happens here. If it keeps happening here and here and here, I'm going to get a chain. Okay, but if we start here, we start septation here, and then all of a sudden maybe septation's here or it's here. Okay, this is what it means to be alternating septation. So if septation's not happening in the same spot each time that the cell divides, we're going to get clusters instead of chains or individual ones. Okay, so like we have bacteria called Staphylococcus. That's because they are cocci shaped and they're arranged in the bunches of grapes. Okay, so a lot of these are very important because we have a specific type of Staphylococcus called Staphylococcus aureus that does the bunches. In fact, we have MRSA, which is methicillin resistant Staphylococcus aureus. This is a strain of Staphylococcus aureus that is resistant to antibiotics, specifically methicillin-based ones. Okay, this is a infection that is acquired at the hospital, and it's very hard to treat because it's resistant to most antibiotics that will treat similar strains of Staphylococcus aureus. Okay, in fact, to treat MRSA, we're going to have to do a long course of the antibiotic doxycycline and the antibiotic clindamycin. So instead of being like penicillin and targeting the cell wall and cell wall production, both doxycycline and clindamycin are going to stop the synthesis of proteins in the bacterial cell. So that's going to be what kills it instead of punching a hole into the side of it like with penicillin and other beta lactams. Okay, I would know what septation means. I would know what alternating septation means, and I would know how you get both rod shapes and cocci shapes during septation. Okay, this will be on your next exam. Okay, so that is 5.4. We talked about the nucleoid, so where the DNA is found. The DNA is looped over itself, and the loops are held constant by DNA binding proteins. Okay, bacterial DNA is found in the cytoplasm, which makes it easy to transcribe it, and then so go from DNA to RNA to proteins. We'll talk more about that in a different chapter. We talked about cell expansion and how it divides into two. We talked about septation and the different ways that bacilli and cocci can get their shapes during and before septation. Okay, so I would know this last bullet point for the exam. It would be very important. Okay, so that is 5.4. We'll see you in chapter 5.5.